backed by popular demand, but with a caveat. So, you guys should only be watching this video if you guys know the parts to protein synthesis, both transcription and translation. Because if you do not, then all of this will just go over your head, as one of my students told me. So make sure that you guys go back to the protein synthesis lecture and watch that first to get all the different vocab, and then come back here to frame your thinking, because this should be a frame of thinking. So we are wanting to know why, right? The big picture of why we are learning protein synthesis. And it's all about that gene expression. We have DNA, and that DNA is usually, and again, this goes for eukaryotes, it's too big and then to leave the nucleus, and we don't want to make all copies of the entire DNA. We just want a section of it, and so we're creating this mRNA that can be used to make a specific protein. So this is the flow of genetics, right? We have DNA to mRNA, which is considered transcription. We're creating a transcript, right? This is the transcript. And then using that transcript and then translating it into a functional protein that express our traits, okay? Characteristics, whatever. But this is the big picture. It's all about gene expression, taking that concept of the DNA and how that DNA is used to make the proteins that, again, express our traits. So let's start at the beginning. I'm going to be using a color that represents my school. And so again, we have transcription. Then, and we're talking about this in terms of eukaryotes, transcription only occurs in the nucleus. Okay, we're going to compare and contrast the eukaryotes to prokaryotes later, but let's focus here. Transcription, the nucleus. So what is happening here? We have a segment of DNA, and we are making mRNA. How's that done? By using RNA polymerase. RNA polymerase reads in the 3' to 5' direction and builds in the, you guessed it, 5' to 3'. So I like this creation of like this formula looking-ish thing, but that's the big picture. And then in eukaryotes again, okay, you only, we actually create something called the pre-mRNA. And so this next step, still in the nucleus, is called post-transcription. That pre-mRNA must be modified. So first things first, a GTP cap, and that's legitimately guanine attached to 3 phosphate groups, is attached to the 5' end of the mRNA. And so again, I'm going to label it here, 5', 3'. Then we have the poly A tail, a lot of adenine at the end, just coding for adenine. So poly A tail attached to the 3' end. And so this happens at the same time here, no true order. And so the third and fourth happens at the same time and is called RNA splicing. So we want to remove the intruders. So these are codes on the mRNA that codes for nothing relevant to the protein that is desired. So introns are removed. And then we have the exons are joined. Again, the process is called RNA splicing. And then we have the third step. So we're labeling, first part is that transcription, post-transcription, and then we have mRNA, and this is the mature mRNA, leaves the nucleus. Let's talk about translation. So again, the mRNA leads to the cytoplasm. Okay, and so the mRNA is going to be used to create that protein with the help of the ribosome. And let's list them all out. And that tRNA. The ribosome is made up of rRNA and proteins. Okay, so let's continue. Let's show all the pieces, the steps. And so again, we have something called the small subunit. And then mRNA attaches to it until it reads that AUG, the start codon. So we have the 5', 3'. So again, start codon. Then with the help of everything else, we have the small subunit, we have the large subunit, and then we have the tRNA, okay, attaching to the codon there. And then we have another, I'm going to draw this one bigger, tRNA with another amino acid. So let's label the parts here. This right here is called the anticodon. Okay, so 3 nucleotide sequences going from the 3' to the 5' end. And remember, everything is directionality here. So this big piece, whoops, forgot to tell you, is the large subunit of the ribosome. Okay, back to this. We have the actual structure of the RNA, so tRNA is in the shape of a T. And then we have the thing attached to it at the very top, the amino acid. Okay, very specific to the amino acid that's being called on by the codon on the mRNA. So this right here is the codon, 3 nucleotide sequence on the mRNA. So now translation is going to be occurring. So this is the step here. It goes in, the bond connecting to the tRNA is broken, and a bond attaching to the new incoming amino acid is connected. Okay, and so once the bond is broken on this tRNA, that tRNA will leave. Okay, now this process occurs over and over again, and quickly drawing it. So this builds and builds and builds. So we have our polypeptide chain, and between each of these spheres, we have the peptide bond, which is considered to be covalent. So this process keeps going. The ribosome is reading it in the 5' to 3' direction. Okay, so it keeps going and going and going until we reach the stop codon. What contains the stop codon? Boom, the release vector. So what happens with that? Boom, right here, y'all. Everything disassembles. We have the small subunit, we have the mRNA, we have the large subunit, and definitely the polypeptide chain folded up to become the desired protein. So that is, again, gene expression, where we take DNA and we make proteins to express our traits. Okay, y'all, that's it for today. I hope this framing helps. Have a good one.